Hi, welcome to Biomindscape. Today we are going to know about transposable element, that is the jumping gene. They are a mobile DNA sequence that can change their position within a genome. They are found in almost all organisms and can contribute to genome evolution and size. They were discovered by Barbara McClinton in Mays in 1940. They are the DNA sequence that can move from one location to another in a genome, and they can be found in prokaryotic bacteria and eukaryotic animal, plant and fungi, and they can exist in multiple copies within a genome. They move through a process called transposition. They can involve either a cut and paste mechanism where the T is removed from its original location and inserted elsewhere, or a replicative mechanism where a copy of T is made and inserted in another place. So they are used in RNA intermediate in the transposition called retrotransposition. So we will know the types. So before knowing the types, we will know why they matter. They can contribute to genomic evolution by introducing new genetic variation. They can insert into gene, potentially disrupting their function and causing mutation. They can be a source of new gene or regulatory sequence. They can contribute to evolution and new traits. So DNA transposon, retrotransposon, line, sign and LU elements. These are the examples of transposive jumping genes. So DNA transposon moved by cut and paste mechanism. It is used in DNA in an enzyme called transposase. Then retrotransposon moved by an RNA intermediate which is reverse transcribed back into the DNA. Then long interspersed nuclear element, line, a common type of retrotransposon found in mainly eukaryotic genome including humans. Then short interspersed element, another type of retrotransposon. It is found associated with line. And LU elements, it is a very abundant type of sign found in human genome. They can be harmful. The impact on health is very important point in learning the jumping genes. So it can also involve in cancer development, and as they can insert into the gene that regulate the cell growth and division, and it can also be beneficial, for example by providing new regulatory sequence that can help gene to be expressed at the right time at the right place. The main function of this is they can cause mutation, alter gene expression and contribute to genomic size variation. In gene expression, genome rearrangement, evolutionary drive, genetic engineering tool, in centromere formation and antibiotic resistance. These are some of the key jumping genes. Mutagenesis. Transposon can disrupt gene by inserting themselves into the coding region, leading to mutation. In gene expression, by inserting into the regulatory region, they can change how genes are expressed. In genome rearrangement, they can cause deletion, insertion, inversion of DNA sequence. In evolutionary drives, transposon, it can contribute to the evolution of the genome by facilitating the movement of recombination of genetic material. And in genetic engineering tool, researchers use transposon to introduce foreign DNA into the genome for research and genetic engineering purpose. In centromere formation, some transposon plays a role in the formation of maintenance of centromere, which are crucial for chromosome segregation. In antibiotic resistance also, in bacteria, they carry the gene from antibiotic resistance, contributing to the spread of antibiotic resistance. So, they are good or bad? They make up a half of the genome in eukaryotic cell, accounting for much of the human genetic diversity. They are also known as selfish gene elements. Many are important in genomic function and evolution. They are very useful to research as a mean to alter the DNA inside a living organism. Let's know the structure of transposable elements. Terminal invertase repeats, that is the short identical sequence at each end of the transposon. Transposon gene encode the enzyme that are cuts and reinsert the transposon. Flanking direction repeats. They are the short sequence creating during insertion into the genome. Now let's know the life cycle of retrotransposon, a genetic element that can copy and paste itself into a new location in the genome through an RNA intermediate. Here are the step-by-step explanation. Retrotransposon DNA is first transcribed into the RNA. RNA is then translated into protein needed for the transposon to replicate itself. The retrotransposon RNA red strand combined with the protein depicted in blue and green here and to form a ribonucleoprotein complex, and these proteins usually include reverse transcriptase and integrase, which are essential for the next step. Within this complex, the RNA is reverse transcribed into the complementary DNA, that is the cDNA, by the reverse transcriptase enzyme. Now the RNA is shown in green. The newly synthesized cDNA, complementary DNA, is integrated into new location in the host genome by the integrase enzyme. This results in a new insert, the gene segment of the retrotransposon in the DNA. The cycle is repeated. Once inserted, the new copy can again be transcribed and the cycle continues, leading to the copy and paste expansion of retrotransposon in the genome. The transposon element can lead to change in gene expression pattern, influencing various cellular processes and phenotypes. In tissue-specific expression also, it can drive tissue-specific expression of gene, contributing to the complexity of gene regulation. It can provide new regulatory element, driving evolutionary innovations and creations of new gene regulatory networks. In disease association also, dysregulation of transposon element has been impacted in various diseases including cancer and neurological disorder. Difference between the retrotransposon and DNA transposon. So a retrotransposon RNA intermediate, it can be used in replication through an RNA intermediate which is then reverse transcribed into DNA. It have reverse transcription. RNA intermediate is converted into DNA by enzyme reverse transcriptase. Then the result is the DNA copy is integrated into a new location in genome. And DNA transposon, it consists of DNA intermediate. DNA transposon move directly from one DNA location to another without an RNA intermediate. Here also cut and paste mechanism is used. The DNA transposon are excised from one location and inserted into another through a cut and paste mechanism. No reverse transcription takes place in DNA transposon. It does not involve. And now the key difference is that the mechanisms of movement. The retrotransposon involve in RNA intermediate and reverse transcription, while DNA transposon involve in direct DNA to DNA transfer. Enzymatic requirements in retrotransposon require reverse transcription, where DNA does not require reverse transposon. The genomic impact in the retrotransposon can lead to increase in the copy number, while in DNA transposon it can result in more precise excision and insertion. So these are the differences between this retrotransposon and DNA transposon, and it is essential for grasping the mechanism of transposable element moving and their impact on genomic evolution. The application in gene tagging. So gene tagging using transposable element is powerful technique in functional genomics to identify a study gene based on the insertional mutagenesis. So it is a technique where the marker known DNA sequence like a transposon is inserted into an organism genome to identify, disrupt and study a gene. So transposon carrying a selectable marker like antibiotic resistance of GFP is introduced in the genome. The transposon insert into random gene, potentially disrupting their functions. There are phenotypic changes which are observed indicating gene function. The flanking sequence, where the transposon inserted a sequence using PCR based method, for example inverse PCR or tail PCR. This sequence is used to identify the disrupted gene by genomic database. So random insertion allow genome wide screening and easy to identify disrupted gene using the known transposon sequence. It is useful in forwarding the genetics, forming a phenotype to gene, and can be used in plants like maize, Arabidopsis, and microbes like yeast and bacteria. For example, in maize, AC and DS, these are the transposon used. In Drosophila, P element is used. In Arabidopsis, activator and dissociation is used. And in yeast, TN3 and T. In bacteria also TN3 and TN5 are used. Going to know about the impact of transposable element on genomic evolution. It is multi-phased and far-reached. Transposon elements have played a significant role in shaping the structure, function, and diversity of the genome across various organisms. Mechanism of the impact: insertional mutagenesis, where it can insert themselves into the gene or regulatory region leading to mutation and alter the gene function. Then gene duplication and deletion, it can mediate gene duplication and deletion event contributing to genomic arrangement. It can provide a new regulatory element such as the promoter, enhancer, and silencer influencing the gene expression. It can facilitate the genomic rearrangement such as inversion, translocation, deletion through their mobility. It contributes in the genomic diversity by introducing new mutation, rearrangement, and regulatory element. It can give rise to new gene or modifying the existing gene leading to the evolutions of a new function. It can rewrite regulatory networks by introducing new regulatory elements or modifying existing ones. And to species divergence by creating a reproductive barrier and influencing the species. In evolution, in human genome, plant genome, and Drosophila genome, it have an importance. So in human genome, transposon TE have contributed significantly to the human genome with many genes and regulatory element derived from the transposon element. It have played in shaping the plant genome, influencing traits such as flower color and disease resistance. In Drosophila also, in the evolution of Drosophila, it influenced the traits such as the wings, the morphology. It can facilitate the adaptation to by the changing environment by introducing a new mutation and regulatory element. It can contribute to the speciation by creating reproductive barrier and influencing the evolution of a new trait.